Stephen Comiskey is one of approximately 9,000 people in Ireland living with MS.
I was first diagnosed with MS about 7 years ago, and I was picked up in the Eye and Ear Hospital where I was working at the time because I developed visual symptoms and blurred vision in my left eye.
What was it like then to get confirmation that it was MS?
Terrifying.
Yeah.
Even though I knew that's what it was, up until that point when all the pieces of the jigsaw were starting to piece together, I knew it was it, and it's only when you get that final diagnosis that it really hits home.
What is MS?
MS is it's a neurological disorder which affects your spinal cord, it affects your nervous system, and it can affect different parts of your body, including your eyes. It can cause different symptoms, including numbness, and it can cause blurriness of vision and fatigue. So at the time when I was being tested for MS, I started to lose feeling in my legs, very cold, pins and needles up to about my waist. I had issues with going to the toilet, and those are some of the symptoms that you can have with MS. Lucky enough, with the type of MS I have, mine got better after a couple of months. It takes a good while for it to go back to normal.
In terms of medication and managing at the moment, what is that like?
So at the moment I'm on an immunosuppressant, and with that immunosuppressant I get bloods taken every 3 months just to monitor my white blood cell count to make sure that my immune system is okay. If my cell count was to drop, then they would take me off the medication and try me on a different type of medication. But there's loads of medications that are specifically to deal with somebody's version of MS or type of MS that wouldn't benefit me. Even if they have the same type of MS as me, they might need a different medication. When I was diagnosed, I'll be perfectly honest with you, I didn't deal with it at the time. I ended up having to go and do a lot of therapy and speak with people. I ended up going to a lot of conferences about MS, and just through that whole process I was able to have a better understanding and really fit MS into my life and not MS become my whole life. But it's made me be more proactive about my health. I recently moved to the coast and I joined up with a local rowing club. Through all these activities, I've actually met people with MS in the community, and I'm able to have those conversations with them now, and basically to reassure one another, you know, it's not the end of the world, and there's always somebody there to help.
Multiple sclerosis is a disease that attacks the central nervous system. This system controls how the body moves by sending signals through nerves in the brain and spinal cord. These nerves are wrapped in a protective membrane called myelin, which helps transmit the signal. In MS, the immune system attacks this myelin, causing a wide range of symptoms. Crucially, it also interferes with the body's natural ability to create new protective membranes. Jessica White is part of a team seeking to reprogram immune cells into helping the body repair myelin.
We're a neuroimmunology lab that looks at how we can boost repair using the immune system. And specifically, we're looking at T cells, and more specifically, regulatory T cells, which are a pro-regenerative subset of T cells. And these have already been shown to boost repair in the central nervous system and could be potentially a therapeutic target for MS patients.
The type of T cells that control how the immune system responds to infection are called Treg cells. They can help prevent attacks on healthy cells and also help to repair damaged cells such as myelin. The goal of the researchers is to enable more T cells to become Treg cells that could help in the fight against MS.
It's very difficult to actually study MS using human tissue, especially the pro-regenerative properties of this. So we isolate spleens from mice, and we can isolate these T cells from these spleens using a specialized kit that essentially labels our cells with an antibody and then also some beads that will magnetically sort out using a magnet.
So you do that one by one?
Essentially, yes.
The ability to isolate T cells is a critical first step. The next stage is to create environments that could help encourage T cells to become a stable population of Treg cells that could then help the body repair myelin. The team at Queen's are working closely on this with their partners in Galway, and I'm meeting Owen McAvoy to find out more. Owen, biomedical engineering is like this rapidly evolving field, but it's also relatively new. Can you tell me a little bit about it?
Yeah, so biomedical engineering is a really exciting field that applies our understanding of engineering to solve problems in medicine. So, for example, how can we generate active forces and how do our muscles work? How does our heart beat? These are all concepts in biomechanics, part of biomedical engineering.
How does that relate to your role in this particular project looking at MS and Treg cells?
We're hoping that we can improve the yield of T cells through mechanical considerations and also to increase their stability so they stay as Tregs for longer and therefore can carry out this role we want in an immunotherapy.
T cells adapt and change depending on the environment they're in. Using simulations, Owen wants to explore the behavior of T cells by designing special environments for them in the lab.
Best way to think about them really is like a spread of jelly, okay? That they're kind of soft, they can absorb water, and you can control the shapes they take based on what environment you put them into. So you can functionalize these hydrogels with different proteins, with different cytokines, different antibodies, and the immune cells will interact with all of these things. You can even pattern them in particular ways if you want the cell to spread out along a line, or in a circle, in a rectangle, even in a star, these complex shapes. And that has a really significant impact on the outcomes for the cell and how it's going to behave, and is it going to become a regulatory T cell.
It sort of sounds like you're building an obstacle course for these Tregs.
Absolutely. In the long term, what we'd like to see is a more effective and a more promising immunotherapy, whereby you can deliver a patient's own cells following some treatment in the lab, some chemical and mechanical intervention, that they can then start to reduce these symptoms of MS, that they can start to remyelinate and improve the overall outcomes for a patient.
It's actually been really nice to have the collaborators over in Galway because they do a lot of techniques like with computational biology that we can't do here at Queen's. So actually being able to bring two sides of biology together has actually really pushed this project far further than we would be able to do alone. And I think that's really nice when it comes to collaborations, that we can utilize two areas of science and drive something even bigger.